Cancer session. And it's my great pleasure to introduce the keynote speaker for today, Dr. Beverly Lyn-Cook. Dr. Beverly Lyn-Cook came to us from the FDA. She started her studies at Fort Valley, Georgia, where she got her a Bachelor of Science. And then she went to Chapel Hill in North Carolina, where she did a postdoctoral training in the Department of Biochemistry, and then went to the FDA after that. And if I'm not mistaken, she started at the FDA in 1988. So I think this year is a momentous achievement of 30 years at the FDA. So congratulations for that. That's big. And we are very much looking forward to your talk. She's going to talk about insights in pancreatic cancer into potential new drug therapy.
Good afternoon. First, I'd like to thank the organizers for inviting me here. This is my first time on your campus. And I always tell my students, I'm very old, but I may look a little young. But I actually spent 10 years at Chapel Hill before going to the FDA. So really I have about 40 years of research experience. What I would like to do today, though, is kind of talk about pancreatic cancer. And if we have any young people in the audience, I'm going to give an overview of the cancer itself to convince you that this is an area of research that you really, really need to consider by the end of my talk. And then at the end, I'll talk about a little of the data we are doing. So hopefully I'll give you some current insights and talk about some of the new drug therapies. Of course, because I work for the FDA, this is my second slide, the views expressed are not those of the FDA. Now, why should we be interested in pancreatic cancer? Pancreatic cancer is the 11th most commonly diagnosed cancer in men and the 9th in women. And it is the 3rd leading cause of cancer death in the U.S. population. And according to the American Cancer Society, it estimates that in the United States, in 2018, 55,440 will be diagnosed with pancreatic cancer, and 44,030 will die from this disease. The expected number of new cases is expected to increase between 2010 and 2030. So this is a cancer where we're going to see an increase. And not only are we going to see an increase, but guess what population we're going to see an increase in? African Americans. So we have to get, this is a very deadly cancer, as you will find out, and we need to get. Now you hear a lot about who has died of pancreatic cancer. You see all the famous people on this slide. But the real truth is, we are dying of pancreatic cancer. The incidence of pancreatic cancer is 50% higher among African Americans than any other racial group in the U.S. And as I said before, it is expected to increase by 55%. So we got to get a grip on this cancer, and we need to educate our people within this area. Why is this cancer so deadly? Mainly because there's no symptoms, no detection tools. So when I talk about strategies at the end, this is what we need the young researchers to start thinking about. And it's always diagnosed at the late stage, stage 4. And then usually the person will die. Surgical resection is the only hope for cure. However, only 20 to 25% of cases are diagnosed at the resectable state. In our case, for African Americans, they don't get to that stage. They don't get a chance to get those resection. For locally advanced metastatic and recurrent pancreatic cancer, chemotherapy is the main treatment, but you'll find out that this is a highly chemo-resistant cancer, therefore the efficacy of the chemotherapy drugs are very limited. Okay, now this is where we're gonna do something about not the non-modifiable risk. These are some of the risk factors, and you heard this throughout today: age, gender. Usually in the past, male higher than women, but guess what? The female have caught up with the males. Is it due to obesity? Is it due to increased smoking? Ethnicity, I just told you African American has the highest incident. Family history, and we know that some of the inherited genetic syndrome can put us at an increased risk of this disease. Now this is a slide I want to spend a little time on. People have been talking about diet and what can we do to prevent these diseases. These are modifiable risks. These are risks that you can do something about. Cigarette smoking. We can do something about that. But guess what is happening? Even with the electronic cigarette, I'm not going to say this, but with the nicotine, you know, we're getting some adverse effect even with the electronic cigarettes. Obesity. Obesity is involved not only in breast cancer, but we are now finding it to be effective in pancreatic cancer. The lack of exercise. The lack of exercise leads to what? Obesity. These are things we can do about it. Diabetes, some association with type 2. Diabetes has been associated as a modifiable risk. Chronic pancreatitis, and that goes back to inflammation. You know, someone was talking about it today, but I also was doing research in my lab on lupus, and inflammation really is going to be the cause for a lot of things, and we really need more research in that area. Occupation exposure, certain pesticides, dyes, chemicals used in metal refining. And here's the one we really can do something about, our diet. We heard about the diet. We have to learn to change our behavior, high-fat diets, diet rich in red meat, and processed meat such as, I know you like it, sausage and bacon. You got to decrease it, all right? So these are things that you yourself can do something about. As I said before, we need biomarkers, early biomarkers, because there's no early detection for this cancer. As you can see on the slide, these are very vague symptoms that you can get. Usually loss of appetite, jaundice is one of the key. Sometimes you have onset diabetes in some of the older individuals. But as you can see, there's no early detection. We have no early markers, so we need early markers for this cancer. Why is this cancer so complex? When we take a look at the pancreas itself, the pancreas is a mixed function organ. That means it has two components. It is an endocrine organ and it's an exocrine organ. So we got to take that into our play when we are talking about how is pancreatic cancer involved. We know that the exocrine part of the pancreas is a key regulator of protein and carbohydrate digestion, as well as the endocrine part plays a role in secretion of insulin for regulator of glucose homeostasis. When we look at pancreatic cancer itself, as we know, this is a typical, early on, we see 95% of our cancers got KRAS, and then as the cancer progresses, you have P16 being inactivated, and then we also have loss of velocity between and inactivation of BRCA2, as well as mutations start to occur in P53. But through genetic technology and through deep sequencing, we know now that there are other passenger mutations that occur along this line as well. And then we're going to talk a little bit about epigenetic changes also occur, where you can gain function, and then you have those genes that you can lose the function through like an activation of some of the suppressor genes. So we know that these genes are involved, and 95% of pancreatic ductal cancer is from the exocrine, and 5% is from the island or the neuroendocrine part of the pancreas. Now, when we look further, we know that pancreatic cancer is a genetic and an epigenetic. We talked a little bit about epigenetic. Epigenetics, those changes that can affect the expression of the gene, not resulting in a mutation. And they usually affect, when you look at over here at the slide, when you see, when I talk about epigenetics, we know that epigenetic involve histone modification, DNA methylation, and also changes in microRNAs. And we're going to find out that what we eat can also modulate all of these components. And when we look further on the complexity of pancreatic cancer itself, we have to look at what are those components in this cancer that makes it so difficult or makes it chemo-resistant. And when we look at the mature and differentiated cell component, but one of the parts that a lot of research is being conducted now is on the cancer stem cell component and also on the stroma of these cells. So we're looking at dysregulation of the microenvironment, which occurs as well as the stromal cells, the immune cells that secrete these factors. So we have to keep in mind the stem cell is really playing a role because they're self-renewal cells, and they are also the ones that lead to the chemo-resistant. And so we are targeting, drugs are being designed to target these stem cell components. But the most complexity about the pancreatic cancer itself is the metastasis and what happens to these stem cells. As the pancreatic cancer stem cells, as you can see, when we look for drug targets, we are looking now at various, how the stem cell can then differentiate and cause the self-renewal. However, these particular pathways has been identified to be critical in this process. So a lot of drugs or potential therapy is being targeted. We are right now working with Dr. Sewall out of the City of Hope, looking at the Wnt signaling pathway, particularly in pancreatic cancer. And we know that the stem cell component and the self-renewal contribute a lot to the resistance to many of the drugs that we're going to talk about later. What are the nutritional requirements? What makes these cells grow? Research has shown that these cells need sugar and these cells need glutamine. And, you know, people say, well, why is this increase in African American population? What are we doing? Sodas, sugar intake. And also we know that glutamine is helpful in progressing the growth of these cells. And we know that also these cells have ability to adapt to an epoxic environment, and they have displayed some autophagy activity for their survival. So these are some of the nutritional requirements that's being conducted now to look at the growth of these cells. But we know that if we can keep the cancer confined to the tumor, then the surgeon can go in and they can section and remove it. But the carpet that behind most of the death in the cancer, of course, is metastasis. So we must understand more of how these cells began to metastasize early on. So we need research in this area. We need research to understand how the microenvironment will play a role in the early stages of metastasis. Now, I want to spend a little time on the challenges and the strategies. What do we need in this field? What type of research? We need to focus on the management. How do we manage if you have the primary tumor? How are you going to manage if you don't have any detection tools to let you know that you have the tumor? Prevention of the metastasis of the cell, and what is the treatment? Now, the challenges comes in here when we say early detection prevention on high-risk population. There's no studies out there. We need studies in this area. We need study exomes from these types of patients to use new types of tools for early detection. This is an area that's a challenge right now. The reason you can't detect the cancer early, because there is no early biomarkers that can be translated to the clinical methods. But you're going to hear about some of the tools later on when one of the speakers is going to be talking about the organoids. I think that is a very nice tool, so you can kind of get some answers to some of these early biomarkers. There is no, we need better prognostic markers, diagnostic markers. The only one FDA has approved for pancreatic cancer is the CA19-9, and it's not very good. It's a good diagnostic marker, but it's not the specificity. It lacks a lot of specificity. The other new tools that we need for preventing metastasis, we have to study the profiles of the tumor itself. And that's when it comes into precision medicine. We need to profile more of the primary tumors and then begin to subtype these pancreatic cancers. They begin to subtype them based on their molecular profile, just like they're doing in breast cancer. Breast cancer, triple negative, have different subtype. Pancreatic cancer can have different subtype based on its molecular profile. So we need that work done. The work is not being done in that area. And of course, the possible treatment regimen, how will you treat the cancer? We definitely need a lot of work in the areas, as you will see. We have very limited drugs to treat this very aggressive cancer. Some of the possible new targets, they're talking about cancer stem cell pathways, the stroma, crosstalk. What is going on? Why is that stroma surrounding that cancer more than any other cancer? And it prevents the chemotherapy drug from reaching the tumors. So now you're targeting the stroma components as well as targeting the pancreatic cancer itself. So treatment, as I said before, surgery offers the best opportunity for long-term survival. About 15% of the cases are diagnosed early. But the thing is, how do you diagnose it early? A lot of times they're caught accidental through imaging analysis, but how many individuals will get that type of screening? As I said before, chemotherapy or chemotherapy with radiation is a choice. And as you know, the golden standard for pancreatic cancer is gemcitabine. And of course, now FDA has approved a combination of gemcitabine with some of the targeted therapy drugs as well. And it seems to be working, but still it's not getting the survival rates that we would like to see. These are some other drugs now being treated being used for pancreatic cancer. But as I said, gemcitabine is the gold standard. And that's one of the drugs that we study a lot in our laboratory. Now, what is the future? What has been done in animal models for targeted therapy? Why do you need targeted therapy? You need targeted therapy if you catch the cancer in the early stage. I mean, that cancer, then that therapy will just target the cancer cell itself. So there are a lot of growth factors inhibitors being looked at, anti-angiogenic factors, drugs have been looked at, and there are drugs now beginning to look at the stroma, targeting that stroma, and as well as drugs that would target the cancer stem cell. So these are some of the classes of targeted therapy that's been looked at. Now, a lot of people talk about immune therapy. Yeah, we are way for that for some of the solid tumors, but believe it or not, some work has been done now on some of these immune therapy in animal models. I think the most success will be with the monoclonal antibodies that will be targeting surface antigens, as well as the cancer vaccines. They are working on those in animal models. As you also mentioned this morning, drugs that target the immune system checkpoints, protein, PD-1, CTLA-4, also are in clinical trials or in animal preclinical models at this time. So these are some of the immune therapy that are beginning to give us some hope for this cancer. But as I said before, until we understand more about early changes that occur in this cancer, that will be more efficient to study this cancer. And of course, CAR-2 cell therapy is being looked at as a way to get the immune system to fight the cancer itself. We've had more success in blood cancers with this type of therapy, but also some of the solid cancers are being looked at as well and being studied. This is what I want to come to, because if we have an increase in this cancer, and diet is playing a role, and we know for prevention reasons that a lot of these dietary components have shown anti-cancer effect. And for years, we've been studying one in our laboratory, endo-3-carbinol, that you can find in broccoli, cabbage, Brussels sprout, those types of family of food. But you can see here, too, a number of other ingredients. So if we have a high-fat diet and we don't eat vegetables, we're not getting these biocomponents that could be very helpful to our body. Now, it's a whole new area of research referred to as nutrigenomics. Nutrigenomics is trying to understand what are these bioactive components in those plants and in those vegetables that I just showed you. How do they affect the DNA, RNA, protein, metabolite? And so a lot of studies have been done with metabolomics, proteomics, nutritional triplomics, and nutritional epigenomics. We have to understand how these biocomponents are working in this sense, because that will give us more on the mechanism of how they can be exerting their anti-cancer effect, or in some cases, endo-3 have anti-inflammatory effects as well. So this is a compound, as I said, that I work with in my lab, endo-3-carbinol, and it's metabolized, as I said, from broccoli and Brussels sprouts and cabbage. And one of the things that we wanted to see is on drug uptake and drug resistance with this particular component, because the pancreatic cancer is highly chemoresistant to a number of drugs, and particularly gemcitabine, depending on the transporter that may be upregulated or downregulated. So what we began to do in our lab is to look at a lot of these ATP-binding proteins that plays a role in resistance. There's a whole 7 subfamily of these genes. We have worked a lot on the ABCA1 and the ABCG. As you can see, these protein binds ATP, and they use energy to hydrolysis to transport molecules. But the critical question that, as a scientist, you want to know is, what is the clinical significance of the transporters in drug exposure, disease, toxicity, and in drug-drug interaction? And what are the characteristics of drugs that can determine whether a transporter may be important or not in their system? And what do we know about the relevance of polymorphism in the various transporters? Because these are critical components. So here, I'm not going to give the data here. I'm going to talk more about the other data, but we did look at ABC transporter and chemo-resistant. This work has been published about 3 years ago. They are very important players. We looked at polymorphism in the ABC transporters, which have been implicated in different response to chemotherapy and survival in various cancer. And we found that a SNP at ABCG2 is associated with overall survival in pancreatic cancer, and ABCB1, C2677T, could predict the side efficacy effect of adjuvant therapy with gemcitabine for post-operative patients. However, what I want to focus on today is human equilibrium nucleotide transporter 1. And it's been shown that this is a high expression of this particular transporter is predicted to predict survival in patients that's treated with gemcitabine for pancreatic cancer. And we actually looked at this with comparison. This is first viability to show you that this is with endo-3-carbinol. Here is a control cell line we use in our lab, HTERT-HPNE, and this is a pancreatic cancer cell line. And as you can see, I3C has no effect on these normal cells. However, I3C can decrease the growth here, but if you combine gemcitabine and I3C, you see a significant decrease in the growth of these cells. And then if we look at the transporter itself, this is the effect of endo-3-carbinol on the expression protein level of this nuclear transporter in 3 different cell lines. And as you can see here, as you increase the concentration of endo-3-carbinol in all of these cell lines, you have an increased expression of the nucleotide transporter 1. And this is the transporter that is responsible for uptake of gemcitabine into the cancer cell. So, and we also looked at this in tissues from cancer patients, and you see different levels of expression of this nucleotide transporter. So those patients, you can stratify the patient as to who will be sensitive to gemcitabine based on the expression level of HEN1. Those who have lower level will not have the capability to be insensitive. They would not be sensitive for the gemcitabine to enter the cells. Now, if you look at the expression level, that with the protein level, we also see the same thing. This is with endo-3-carbinol by itself. So a dietary component can upregulate this important drug transporter. This is something, one in which that can then make those cells sensitive to gemcitabine. Not with even using gemcitabine by itself. This is just with endo-3-carbinol. And then when we looked at the combined effect, of course, for this transporter, we saw similar results. This work was published about 2 years ago. You can see with the combination, you still get a significant increase, but we saw also you got an increase without using the drug itself in all 3 of these pancreatic cancer cell lines. But this is 4 of the cancer cell line. Some responded differently than others. You had the highest expression in the PAN1 as well as in the MIR cells, PAN2. And also, I3C increases gemcitabine cytotoxicity in these cells. And again, this is on several of these cell lines. You can see that gemcitabine plus I3C decreases decrease the growth of these cells tremendously, and compared to gemcitabine alone or in the combination of an inhibitor. So I3C increases gemcitabine cytotoxicity in pancreatic cancer cell itself. But remember, it did nothing to normal cell. So it's targeting the cancer cell itself. So the challenge remains with us is that where do we go from here? How do we translate all the information that's been received, even for pancreatic cancer or other cancer, into information that can be useful for the consumer, for the FDA, for drug labeling, as well as for clinical practice? And one of the things that we have learned over the years, particularly in drugs, and this is whether you're looking at sex difference in response to drugs or chemotherapy, particular chemotherapy drugs, because they have such a narrow therapeutic index, we have found that one size does not fit all, and that until we understand the genetic background of these very tumors and begin to subtype them based on their molecular profiling, just like we're doing for breast cancer, we'll be able to determine who will be more vulnerable and who will be able to respond to these chemotherapy drugs. And so that brings us to personalized cancer therapy. That's where we want to go eventually. So, and I think we're there with the Moonshot Initiative and with the research being conducted now with molecular profiling here with the patient. But the problem with African American is the sample size. There's not a lot of African American pancreatic tumors out there because, as I stated before, they did not get surgery. They died, okay? So we have to, and a large effort is being done now, probably here also working with other groups and trying to obtain large sample size of these pancreatic cancer cells, particularly from African American, so that they can be profiled. And by profiling them, hopefully we will come up with better prognostic markers and markers that will predict the sensitivity and the resistance of the drug, as well as what we are more concerned about in the FDA, markers are predictive of adverse effect or more severe side effect. And then we'll be able to then develop drug therapy based on individual response and based on the molecular profile of the tumor itself. So I think that's the way we have to go, but we also have to think about prevention strategy, as I stated before. If the increase is on the rise in African American, do they know? Is the word been put in the community? We talked about a lot of community work this morning, and I didn't hear too much about pancreatic cancer. And the statistic is there. It's like it's a little secret that no one want to talk about. But it's something that we really have to let people know, because I've known 3 people to die of pancreatic cancer in the last 8 months. And they were all African American, and they had no symptoms until the end. So this is an area that I would like to, if you're not doing research in this area, I'd like to encourage you to do research in this area. There's grant money, there's a lot of grant initiative being pushed toward pancreatic cancer, and it's a very nasty cancer once you get it, and it's very deadly. So hopefully this is kind of enlighten you just to think about pancreatic cancer, to think about those modifiable risks if you're doing some of these things. Change your behavior in order to be helpful. So I'm going to stop here and acknowledge my people in the lab. I didn't get a chance to talk about just one person work, Hogan Wayne, who is now at CEDAR in Washington, one of our centers in Washington. He was my commissioned fellow and did a lot of the work. Lee Payne did a lot of the work that I didn't show on the polymorphism. That work is published. And then I just want to mention one of my postdocs, Nancy Joseph, who's a graduate of your institution here. She's a SUNY. She's a graduate of SUNY, and she did a lot of work also with pancreatic cancer and metformin. A paper just going to be coming out soon on that work, looking at not only the nucleoside 1, but also the nucleoside 4, and its involvement in the efficacy of those drugs in combination. Thank you.